Okay, so you watched the two videos on antigen presentation, and the main difference between the MHC1 and the MHC2 is that MHC2 would be specific to, for example, the macrophage and other APC cells, antigen presenting cells. So when they do their phagocytosis, then they display those pathogen epitopes with MHC2. MHC1 would be presented with what we call endogenous antigens, things that are self, or also MHC1 presents antigens from virus. But viruses are inside the cell, so those are also presented with MHC1. All right, so let's look at T cells, because that's really the next step. Once the antigen presenting cells present their antigens, they're kind of dancing around with these little epitopes from the pathogen on their surface. Then they interact with what are called helper T cells. So helper T cells are, let's see if I can get a pen going here, sometimes called TH, that stands for helper T cells. Cytotoxic T cells would be called TC. But anyway, let's talk about helper T cells. What they do, helper T cells have on their surface what we call a T cell receptor. T cell receptor. And the T cell receptor has various shapes, and your body makes thousands and thousands of different helper T cells, and each of them has a different shape on its T cell receptor. So T cell receptor is a transmembrane protein that can bind to, or we say recognize, an epitope. So this would be the T cell. Let's say this is the helper T. And it has this little receptor, but there's different ones. Your body makes different ones. I don't know why my mouse always does that, but it does for some reason. So this would be a different helper T cell, and maybe it has a receptor that isn't a triangle, maybe it's a square. And then there's another helper T cell that, and this is very simplistic, but this one has a circle. So these are like locks. You can think of these as like locks. Like a lock and a key kind of concept. So the T cell receptor is kind of like a lock. The key will be whatever's on the antigen presenting cell. So here's an antigen presenting cell. And let's say that it is presenting an antigen that is a triangle shape. So the pathogen has this antigen, and here's the antigen on the surface of the APC. Then each of these cells will come up, and so this is kind of like the key. If we're using this analogy of the lock and the key. So each of these T helper cells tries to put its lock on the key. And you can see that this one, the first one, would fit. These ones don't fit, so they just, they just go away and they're sad. But this one fits, so that activates when it fits, when this receptor is attached onto the epitope, the presented antigen, then that activates a reaction in which the APC stimulates this particular T helper cell to proliferate, cell division. So you get thousands and thousands of identical copies of this T helper cell particularly. What this means is that your body has thousands of different T helper cells, meaning thousands of different receptors, before the pathogen even comes into the body, which is really amazing. But there's only really like one of each. Well, I don't know if it's exactly one, but there's very small numbers of each of them, but the one that fits, the one that reacts to that particular pathogen, will be stimulated. The stimulation is a chemical called interleukin-1, IL-1. And that is released by the APC and stimulates that T helper cell to proliferate. So then you have thousands of T helper cells that have the correct T cell receptor that matches up with the antigen that was presented. So that's really the step here that involves the T cells. So the APC interacts with the T helper cell. All right. Then the T helper cell is going to then activate the next group of cells. So APC processes the antigen, displays it with MHC2. The correct T helper cell is then activated, and then we'll get to the next step. So that's antigen presentation, step 1, T helper cell activation, step 2, and then step 3 is coming. Step 3 is going to involve the B cells and the cytotoxic T cells. The B cells are going to produce antibodies, so that's your humoral response. And the cytotoxic T cells are going to act very much like the natural killer cells, the NK cells, and they're going to do killing. They're going to be activated to do more killing, but the B cells, the antibodies, the release of antibodies is going to be coming next. So let's stop this video. I think it's a good time. Stop the video and watch this little animation. I found that the animation doesn't load like immediately for some reason. I don't know why. Let me see if I can get it to load. But you got to be patient with it. Yeah, it looks like it's fine now. But anyway, I'm not gonna show it right now, but you can stop and watch this. It's on eCampus. And I think that's really going to help you understand this second step. But I'm going to move on to the third step. Okay, so you've watched the video of how the T helper cells are stimulated. And the T helper cells will then stimulate the B cells, and we will talk about the B cells. I do want to talk, though, about how does the body prevent stimulation of T helper cells that might react against self. And the way it works is that the body has a system called clonal deletion. Clonal deletion eliminates any self-reactive T cells or B cells, although we're talking about T cells right now, but the system is very similar for the B cells. For the T cells, this is going to happen in the thymus. Any T cell that reacts to self-antigens, specifically the ones presented on MHC1, or any T cells that do not correctly recognize MHC, which is an important part of this, they will undergo what's called apoptosis. And apoptosis is cell suicide. So cell suicide, apoptosis is cell suicide. That's how I first learned it. And then as I got further along in my education, they started using the language programmed cell death. But these mean the same thing. Just sounds a little more fancy. Programmed cell death. Like an intentional death. So the cell kills itself. So any cell that is self-reactive kills itself. And what that does is prevents an autoimmune disease, because an autoimmune disease is where you have a lymphocyte that is self-reactive, and so when it detects self, it stimulates killing and production of antibodies, and that targets your own cells are then killed, which is, causes issues, causes problems. So, but let's assume this goes properly. T cells that don't react to self are left. Ones that do react to self die, the other ones are left. And so that presumably those would be the T cells that potentially can react to any foreign thing that comes into your body. And those are your protective T cells. All right, let's talk about B cells, because we said that once the proper T cell, helper cell, is activated, then it's going to activate the correct B cell. Just like the T cells have T cell receptors, B cells have B cell receptors. And just like T cell, there's thousands of different kinds of T cells with different T cell receptors. There's thousands of different B cells with B cell receptors that are all different. And each B cell has just one B cell receptor type, and that recognizes a particular epitope. So again, you have all these cells already set up in your system. I think it's amazing. And each cell would have its particular B cell receptor. Of course, there's not just one on the surface, but I'm just drawing one. They would be all over the surface, but I'm just drawing one. Okay, so very similar to the way it was with T cell receptors. So these are your B cells. And again, the correct antigen, or sorry, not the correct antigen, the antigen, let's say this is the APC, then you have the antigen here, so we'll go with the triangle again. So if this antigen is this, then this B cell would be activated, just like the corresponding T cell would be activated, and this will proliferate. But the B cell needs the T cell to help with the activation. But anyway, these ones would not be activated. They don't die, they just don't proliferate. And so the one of them proliferates, you get thousands and thousands and thousands of these guys, all having the same receptor, which is specific for the current epitope. All right, now that when the B cell is activated, what happens? Well, what it's going to do is the B cell receptor is actually the same thing, the same molecule as the antibody. The only difference is B cell receptors are on the membrane, they're attached to the membrane, and once this B cell gets activated, then it will start secreting its B cell receptor into the serum. It'll release it into the serum. It'll make more and release it. It doesn't release the one that's already on its surface. It makes more of that, and then it just releases it completely out of the cell. So antibodies are fully secreted proteins. B cell receptors are attached to the B cell membrane. That's really the difference between them. So what does an antibody look like? And really, what does a B cell receptor look like? First thing you should know is that antibodies are the same thing as immunoglobulins. All right, Ig, sometimes we use the abbreviation Ig for immunoglobulin, but those are antibodies. The activated B cell, we kind of changed its name. Sometimes we'll call it a plasma cell. That's an activated B cell. It's been activated to secrete its particular kind of B cell receptor. So it's secreting this as an antibody into the serum portion of the blood. The structure of an antibody, it has 4 protein chains. This is a protein chain, and this is a protein chain. These are called heavy chains mainly because they're the longer ones. And the shorter ones are called light chains because they're shorter, they're lighter. And then this is all connected through these covalent bonds called disulfide bridges. And if this was a B cell receptor, then this part of the B cell receptor would be embedded in the membrane here. If it was an antibody, then the whole thing is just in the plasma. But I kind of, on the last slide, I didn't really show it completely correctly, because an antibody, it always has this sort of V shape, but where the antigen, where the epitope would bind is right here at the end here. And so each antibody can actually attach to two different, not two different epitopes, two of the same epitopes, but on two different pathogens. Like this could be, like there could be a bacterial cell over here that has that little triangular epitope on it, and there could be another bacterial cell over here that has its triangular epitope. So each antibody will attach to potentially two different pathogens. It's also possible for it to attach to two epitopes on the same bacterial cell. That's possible too. But right here at the end, this is where the antigen will be attached. So when we talked about the lock and the key, there's actually two locks. There's a triangular shape cutout on this one and a triangular shape cutout on this one. Or if we were talking about the square one, a square here and a square here. So there's actually two binding sites on each antibody. Earlier I kind of just showed one. But in any case, each plasma cell secretes just one particular kind of antibody, but it's the one that has been selected through this process. So it's the correct antibody for the particular pathogen. This part of the chain is called the fraction crystallizable, which is kind of complicated, but think about the C standing for the constant. This part of the chain is always the same. Every B cell has the same proteins in this section. The only part that's different between the different B cell types would be what's up here in this particular antigen binding site. So the protein would be different for the antibody that attaches to the triangle epitope compared to the antibody that attaches to the square epitope, if you know what I mean. But the rest of this would be consistent no matter what antibody it was or what antigen it reacted to. So what do the antibodies do? Once the B cell is activated to release the correct antibodies, there's really 5 functions, but I want you to focus on these 3 here. If this is an introduction for you, if you know a little bit more about antibodies, you can take a look at these. But I really think that starting out, these concepts are very helpful. All right, so the first thing is called neutralization. Remember that each antibody has 2 binding sites. And so what you can end up with is a whole bunch of antibodies of that type are just kind of attached all the way around, if it's a bacterium or if it's a virus or if it's just something else. And really what they're doing is they're just blocking that virus or that bacterium from being able to do what it wants to do. Bacteria usually want to bind to other surfaces. So this bacterium would have trouble because the antibodies are in the way. It's blocking. The virus would not be able to enter its host cell because the antibodies are blocking it. In this picture, it's very two-dimensional, but you can imagine those antibodies all around the surface of the virus in 3 dimensions, kind of creating this complete coverage on the surface. So it really blocks that pathogen from being able to do anything. It's just blocked. And so we call that neutralization. Not completely unrelated, but a different term that we use is called opsonization. So once a bacterium, let's say, is covered in antibodies, what that tends to do, opsonization means to make tasty. And the first time I was taught this, I thought that was kind of a funny definition, but it is the definition, to make tasty. So this here is a phagocyte, an APC cell, let's say. And APC cells will take in lots of things, but they really find pathogens surrounded by antibodies. They find them even more delicious than pathogens that don't have any antibodies. So it makes it it increases the intake of the pathogen into the APC cells if they are covered in antibodies. So opsonization, it increases the phagocytosis of that pathogen. And then agglutination is where, remember, I said that each antibody can attach to 2 different pathogens. So it kind of ends up, you can end up clumping up the bacteria in this picture, kind of clump up a little bit, and that sometimes helps. It really kind of neutralizes and opsonizes at the same time, but the agglutination is kind of this clumping thing. And this can happen in the body, but also this is the actual reaction that we're looking for outside the body when we do just a serological test, kind of this agglutination thing. But inside the body, it kind of neutralizes and increases opsonization as well. But agglutination specifically refers to the kind of the clumping of things. It helps to attract phagocytes. So antibodies are really important to these functions. One of the other things that antibodies do is stimulates complement. You may remember when we talked about complement, I didn't make a big mention of it, but complement will actually antibodies will really attract complement. And complement makes those membrane attack complexes that create holes in the surface of the pathogen. So there's lots of things that antibodies do. So this is another video that kind of goes over what I just talked about, but I think it's helpful to see it in action, the functions of antibodies. So take a minute and look at this video. But I'm going to move on to the different types of B cells. There's 5 different types not the different types of B cells, excuse me, the different types of antibodies. There's really 5 different types of antibodies. And you have IgM, IgG, IgA, IgE, IgD. Some of these are more involved with allergies. IgA is more something that would be in your body secretions. I'm going to highlight these 2 since we're just introducing these concepts, because these would be the top 2 that you would start with. So I'm not going to dig down too much into the detail here, but we're going to look at IgM and IgG. IgM is kind of awesome if you look at it. IgM is actually 5 antibody molecules that are all attached to each other in this really cool kind of wheel looking structure, kind of this round structure. I don't know what wheel, kind of a wheel. So it's actually 5 different ones, but they're all attached covalently. So IgM is actually usually the first antibody that's made. If you look back on your images of complement, this is the one that's involved in complement as well. So that's the first one that's made, and it has so many of the binding sites because each of these has 2, so it's 10 binding sites on each IgM. So it's really going to be able to find those antigens in the body. That's the first one produced. Then your body switches and starts making and releasing just IgG. So IgG is the most common. And by the way, these are Greek letters. So this is mu, gamma, alpha, epsilon, delta. So sometimes we talk about gamma globulin. That's IgG. So, but anyway, if you ever heard of that, it might make sense. But anyway, doesn't matter. We're going to call it IgM, IgG. And IgG just looks like the way I presented a regular antibody to look. IgM is the one that's kind of cool. The rest of them basically look like this. Some of the secretory forms have 2 attached to each other, but it's not hugely different. But IgM is just big. But anyway, IgG is usually produced second. It is the longest lasting of the antibodies, and it's the one that you would most commonly find in the blood. So when you're first exposed to a new antigen, your body will make IgM, and then later start switching to IgG. Now, what difference does that make? Nothing in terms of how the antibodies function, but what it does do is if you can tell which antibody is being made in a patient, you can tell whether they're having an initial response to it or whether they're having a second response to it. So we'll go over that in the next slide. The antibodies themselves just do what all antibodies do: neutralization, opsonization, agglutination. But because there's different types, you can actually figure stuff out by figuring out which types of antibodies are being made in the patient. But IgM has this structure. IgG is more typical. So those are your antibodies. Now, just like T cells go through clonal deletion, B cells also go through clonal deletion. They go through it in the bone marrow, and we're not going to dig down too deep into this, but just like before, any B cell that is autoreactive will die. So they're showing you here's a B cell that's autoreactive will die. B cells that are not autoreactive are allowed to live. And again, this prevents autoimmune disease, which is where the body reacts to self. So apoptosis, again, is what happens when a B cell reacts to self. Okay, but let's talk about these different types of immunoglobulins. Let's say that you have contracted a pathogen, like you've been infected by something and it's growing. It can be bacterium, it can be virus, or you've been given a vaccine, which is like dead virus or dead bacteria. Your body doesn't know the difference between dead or alive. So a dead bacterium or a live bacterium or a dead virus or a live virus will cause the same response. Your APCs will process the antigens, present them to the T helper cells. T helper cells will then stimulate the correct B cells to make the antibodies. At first, when very first time you've ever had that pathogen in your body will make some IgM. Now look at this. This is, day 6 or something like that. Maybe day 8. There's a lag. It takes a while for your body. Now this is not this is not true of everything. Some things take longer than others, but something like this. Like in the first week, you've got some IgM that's being produced typically. By the way, some antigens create much stronger response than others. I think I mentioned in the very first part of the lecture that large molecules typically because they have more epitopes, they will create more reaction. So you might get a stronger response, but that doesn't matter. But you'll get some kind of IgM release, in the first week or so. After, somewhere around the second week, you'll stop getting the IgM and you'll start getting IgG. Now that's your first response. But what's important here is that even though the cells that are making the IgG stop making it, they become memory cells. So you have these memory cells that are left. They're not making the antibody anymore, but they're there. And they're alive. And they stay in your blood. So the memory cells are the ones that are particularly making that particular antibody type. So let's say you get exposed again. This could be a second time or a third time or a fourth time. Anytime after the initial exposure, what will happen is, look at the timeline here. Day 6 over here. So here on day 6 or so, we were just seeing the first IgM production. But when it's a second response, then there's not very much lag time because those memory cells are already there and they'll just start making that antibody quickly. So now we're on day 3, for example. So the IgM response is about the same level as it was in the initial response, but it's much faster this time. What is much different, though, is the IgG response, because now the IgG response is not only much faster, it's also much higher. So you get IgG about the same time as you get IgM, and you get tons of it, and it stays. So the secondary response is the much stronger response. That's why you want to vaccinate, because when you vaccinate, you allow somebody to go through this whole process without getting sick, so that when they really get exposed to the real virus or the real pathogen, they're going to react immediately and very strong. So that's why vaccination really works, because it sets you up without actually getting the disease. It sets up your immune system as if you had had the disease, so that when you really do get exposed to the real thing, your body's all ready to react quickly and with a high response. So those are the things you need to look at. So if we measured somebody's IgM and IgG, we could kind of tell, we might be able to tell if this is their primary response or their secondary response, depending on how those numbers look. And we would look at how high the numbers are to see how much of a response their body is making. The last topic in this chapter is really vocabulary. You get immunity either naturally, that's when you get the disease, you actually get through the disease, you get the actual live virus or the live pathogen, we'll call it live. We talked about virus can't be really live, but you get the live pathogen, you get sick probably, maybe you're asymptomatic, that's possible too, but you recover. If you recover, then you have naturally now acquired that immunity. You now have those memory cells, so that when if you get it again, you'll have a faster response to fight it off. Artificially acquired immunity means you didn't actually get the live pathogen, but somehow it got into your body, like injected with a needle. So a vaccine would lead to an artificially acquired immunity. Now, both of them are fine. One's not better than the other. In fact, a vaccination doesn't put you at risk. You get the immunity, but you don't, because there's a chance, in terms of this naturally acquired immunity, there's a chance you won't recover. So some people might not survive. So artificially acquired is a lot of times better, because you're not suffering the disease. You're just getting the dead pathogen, so your body can set up the reaction. The other terms that are used are active or passive. Active means your immune system makes the antibodies. Your own immune system makes the antibodies. So even if you get a vaccination through an injection of dead pathogen, it's still your immune system that makes the reaction, makes the antibodies. And when you have an active immunity, when you set up an active immunity, it means you'll have memory cells. Makes the antibodies, AB, I'm going to say antibodies, and has memory cells. There is also something called passive immunity. Passive immunity, though, there's no memory cells. Your immune system didn't make the antibodies. If I injected you with antibodies, injection, then because it's an injection, it's artificially acquired, and if it's antibodies that your own body didn't make, but maybe they were made in a laboratory, then your body didn't make them. You don't have memory cells for them, just the antibodies, just the proteins were injected into you. That's called passive. Passive immunity is not very strong, and it's kind of short-lived. So you can get an injected antibody that would be artificially acquired and passive. So sometimes people who have immune deficiency get injections of antibodies, but they have to keep going to the hospital and getting the injections because they don't have any memory cells that can live long term. They have to keep getting more injections. There is, though, sometimes you can get a passive immunity that is natural, and that would be, the main example of that would be breast milk. Breast milk, it's a passive, the baby is getting a passive immunity because they're getting antibodies through the breast milk, but the breast milk antibodies are considered natural. It's not an injection. So usually the definition of natural and artificial, in the end, if there's a needle involved, then it's artificial. So breast milk naturally has antibodies, but the baby is getting them in a passive way. Their body doesn't have memory cells. All right, so naturally acquired, you actually get the disease, you actively have it, you have memory. That'll be a very strong immune response, assuming that you survive, depending on what the disease is. Artificially acquired but active would be a vaccination. So a vaccination. Passive but natural would be like antibodies in breast milk for the baby. Passive and artificially acquired would be, this is pretty specific, it would be injection of antibody for somebody who has an immune problem. So mostly it's just a terminology thing that you need to make sure you're very clear about. All right, that's the end of the lecture for Chapter 16.